Hello. Today I will be presenting TIMEOR, Trajectory, Inference and Mechanism Exploration with Omics data in R. This is the first automated interactive web and command line time series multi-omics pipeline method for differential expression and comprehensive downstream analysis. Overall, TIMEOR reconstructs an interpretable and user-guided gene regulatory network, and specifically, TIMEOR's web interface leverages the user's knowledge of the biological process by providing options to users after answering 6 questions. During each analysis phase, TIMEOR takes the raw sequenced reads through FASTQ files that you can extract through Geo and performs all analysis from quality control to differential expression and enrichment, and then TIMEOR integrates motif and ChIP-seq data to define transcription factor binding patterns and reconstructs a transcription factor gene regulatory network. This is all done in the pre-processing, then primary analysis during the differential expression phase, and enrichment, factor binding, and GRN or gene regulatory network construction is done in the secondary analysis phase. This web interface enhances the reproducibility and ease for users to dedicate more time to interpreting results and planning follow-up experiments. So overall, I would say that this is a great adaptive method to predict these gene regulatory networks from time series RNA-seq, which integrates motif enrichment, ChIP-seq data, and temporal gene interactions. Another important thing to note is that TIMEOR will be hosted at the DRSC TRIP core facility at the Harvard Medical School Research Computing Center. This will be a very good way to maintain TIMEOR and to ensure that multiple users can access TIMEOR's capability all at once. So today I'm going to be speaking with you about the interface. So first, we start with processing raw data. We're going to go to Geo and search for a time series RNA-seq data set. So I have one set out already. This is the one used in the original paper. And we're just going to extract 2 samples to show you. As it says, we want to click on the sRun selector. In this case, we can also see that in the top here, run selector and go. Then we can download the data or select replicates by clicking on the metadata under select. So here, what we're going to do, I'm just going to grab the 2 small ones, metadata, and this is the SRA run table that is output. This is going to be processed and a metadata file is going to be created from these data. So now that we have that, we can set the input to either the SRA run table, which we just downloaded, or a pre-made metadata file, and we'll get into that in a second. So this would be the metadata. We have an SRA run table, and then you can upload and choose the SRA run table and click open. When this is done, what you'll want to do is determine the adaptive default parameters. So the first will enable you to determine the organism, Drosophila, Homo sapiens, or Mus musculus, the type of sequencing, paired end or single end, what type of experiment, case versus control, or just case or control, what type of time series, close time point and long time series, close time point and short time series, and distant time point, and either comparing multiple methods at the alignment and differential expression stage, specifically using a splice site aware method such as HiSat2 versus bowtie 2, and the differential expression methods, which are both categorical and continuous, so specifically DE-Seq2, Impulse DE2, and NextMagSig Pro. So we're going to say yes. And then here, what are the maximum number of time steps over which one gene can influence the transcription of another gene? So what we're asking here is if there is a window of interaction that you would like to consider all possible interactions in. And then you can click Run. So these steps have been provided for you specifically in the example data, loading real data, where we take the SRA run table of the 2 replicates that I previously showed. So you can just click here, and what will happen is that that same SRA run table will be converted to a metadata file, and you see that here. It will also create an informative ID versus just the SRR ID here. This is a bit more informative. The adaptive default methods that we just went through will be set. Then we can press Run to process the raw RNA-seq time series data. And note that this example is just going to explain the Process Raw Data tab. We have another example that runs quite quickly for the next steps. So by clicking Run, the raw data are retrieved, quality checked, aligned, and saved. So what we see here is that the data have been retrieved, the quality control has been done, and we see that here. This is a summary. We have our 2 samples or replicates. We have our percent duplicates, the percent GC content, the average sequence lengths, the percent of failed reads, and the total sequences. We can download an interactive results summary as well. This is done using MultiQC. They have a very nice interface that we can interact with to check all of the qualities for these reads. Next, the data are aligned. I clicked yes to comparing multiple alignment methods. So if we scroll down, we'll see that HiSat2 and bowtie 2 have been run. The user can then decide which method they want to use in order to then generate the count matrix. So here I'm going to pick bowtie 2 as it has an overall higher alignment rate. It's just an example here. And then you can click count, generate count matrix. Great. This count matrix is then passed to the next stage. Now we can use the simulated data to describe the rest of these sections. This is done in order to highlight the additional kind of data input that is allowed. So if you notice right here, if you'd like to process your own data, TIMEOR allows you to jump to step 4, which is essentially to load a pre-metadata file, and then to go to this tab and load your own count matrix. That means that the data have already been processed in the way that I just described, and you would like to perform normalization, then the steps in primary analysis followed by secondary analysis. So in order to do this, the example is already provided to describe the second way of inputting your own data. In this case, the simulated data have been uploaded, so that is the metadata file, the adaptive defaults have been set, and a count matrix has been loaded, and we're going to see the Load Count Matrix tab. And then we can see how to normalize and correct these data. This is the count matrix. These are interactive plots showing you the replicate correlations. You can download a plot of any part of the graph that you're interested in. You can also look at the Pearson correlation instead of the Spearman. And again, you can also pan, zoom in, zoom out, and reset. The same is true for the PCA plot. Given that these are the first two principal components, you can also look at the third, fourth, and so on principal components. Let's see if we can normalize and correct to increase the percent of variance explained here. So the user can choose between several normalization methods. The correction method here is Harman. We do have some improvement. And again, we can see the Spearman correlation or the Pearson correlation if we so choose to between our replicates. We can also check another method, this upper quartile, to see if we can have a better normalization and correction. And indeed, we do see a better principal component 1, so variance explained. Using these normalized and corrected data, we can now move on to primary analysis. Here, what we ask the users is to name the results folder. Because I selected to compare multiple methods, TIMEOR is going to run Impulse DE2, NextMagSig Pro, and the categorical method, which is not normally used for time series data, but has proven to be useful, is DESeq2. We've named the folder, and we've named the we've set the adjusted p-value threshold. We can now click run. TIMEOR is asking us to click the render Venn diagram to see how these methods compare in terms of their results. When we click render Venn diagram, we can see that there is a high overlap between Impulse DE2 and DESeq2. There's also a complete overlap with NextMagSig Pro and Impulse DE2, and we can also consider adding a previous study, which is very useful if we have used data from another study or if we are comparing with a previous study. When we do that, we have an additional overlap here of the previous study. We're going to continue with Impulse DE2 results, given that it has the highest overlap with the previous study and with other methods. And we can see that here. The user can display whatever method results he or she chooses. Here we're displaying Impulse DE2 results. And these results can be then clustered in a cluster map. TIMEOR automatically clusters the gene trajectories. You can also choose as the user to manually determine the number of clusters. Here in this example, we've allowed TIMEOR to automatically designate 3 clusters. And again, we can zoom in and take pictures as we see fit. Maybe we want to take pictures of each of the clusters. We can pan and see the genes. Okay. Once we have chosen the resulting differential expression method that we want, as well as the clustering that we choose, we can move on to secondary analysis. In secondary analysis, we first will see an enrichment tab followed by factor binding, and then determining temporal relations with these data in order to determine the gene regulatory network. So here we see the cluster map from before, and we can start to interact and look for enrichment in each of these 3 clusters. The names of all of the genes in that cluster appear in the color of that cluster on the right. They are listed in this way in the event that the user would want to copy and paste this list into some other tool. So that's useful for the user. And then once we determine what cluster we would like to analyze first, we can click Analyze On, and TIMEOR will run enrichment for Go terms in molecular function, biological process, and cellular component. It will also perform pathway enrichment and network enrichment. And it will also perform de novo motif analysis to identify the top 3 de novo motifs. You can also download the meme interactive results if you so choose. And here we can observe the output of meme and where those motifs are. This can be done for all of the clusters. You simply click Analyze Off to remove the current results, and you can continue with the other clusters. Now we can move on to factor binding. On this tab, what you will observe is the cluster map, as before. You will also see that TIMEOR has identified the observed or perturbed transcription factors in each of these clusters, as well as predicted transcription factors that could be binding to an enriched set of genes, or is enriched to bind to a set of genes in each of these clusters. And it does so by leveraging RSys Target's ability to run many transcription factor binding methods, and I will show those details in a moment. And each of those methods has a ranking for the top, in this case, 4 transcription factors that are putatively binding to each of these clusters. If at least 40% of the methods agree on the top predicted transcription factors, those are reported, as you can see here. So as an example, the top transcription factor that is putatively binding to cluster 2 is this STAT92E. If we'd like to look at the results, we can move down, click on 2, and then see which methods actually output the transcription factors. So we can then also see, for this example, I set the threshold lower, given that this is a simulated data set. If there are blanks here, that indicates that there is an enriched motif that hasn't been assigned to a transcription factor region. So in that case, we can download the interactive cluster motif results and visualize those for the user. And you can filter as you see fit. And actually see those motifs. Each predicted transcription factor that has any chip data has its associated encode ID that has been extracted using TIMEOR. The user can then take these IDs and move to the next step to load bigwig files in order to visualize transcription factor binding across all of the clusters. So As an example, I will show you what we do. You'll go to ENCODE, take the ID of interest, search for that bigwig file, and then upload that bigwig file to TIMEOR. So I will start with one here. I've downloaded several bigwig files already. Once the bigwig file is uploaded, you just click go, and it will produce, TIMEOR will produce the average profile over each cluster for that particular transcription factor. And the colors are associated with the clusters. Next, on the temporal relations tab, we again see the gene expression trajectory clusters. We also see the observed and top predicted transcription factors. This list is by orthology that they have been determined. Then what TIMEOR does is integrates the set of transcription factors across the clusters and how they interact, both the observed and the top one transcription factor. The temporal relations table takes this information of the temporal dynamics for each cluster and the observed and top predicted transcription factors, either in that cluster or predicted to bind to genes in this cluster, an enriched set of genes in each cluster, and shows the temporal relationships between the observed and predicted transcription factors. These two together enable the user to visualize the gene regulatory network of their interest. And in order to facilitate the user in his or her ability to express what's happening from the experiment, TIMEOR has integrated string DB so that the user can move these nodes around and describe the relationships as they see fit. So to describe this table a little bit, the four types of relationships that are shown are the regulator, in this case the observed transcription factor, having a known either activation or repression effect on another observed transcription factor. The next relationship that is, and you will see observed known interaction, in this case it's activated. Another known relationship that you observe here is that these observed transcription factors can have a predicted interaction, meaning that they do not have a known interaction that has been experimentally determined, such as HEY and ERC, but given that they are in this network together and they show temporal structure that might indicate that this transcription factor is temporally interacting with ERC in this case, TIMEOR will report a predicted interaction within this time window. The other two types of interactions that can be observed are between the predicted transcription factors acting either in a repressive or activating state on the observed transcription factor. So the way that this relationship works is that the predicted transcription factor will have some effect on the observed transcription factor, either activating or repressing that transcription factor. And then when there is a predicted interaction, meaning that there has not yet been an experimentally determined interaction between these two factors, such as STAT 92E and OVO, that one can be predicted and tested All of these relationships can be identified and depicted as the user sees fit by simply typing in whatever genes the user wants into this search, clicking search, and following the StringDB steps in order to essentially move these genes around and add any kinds of interaction edges that best describe the gene regulatory network. There is a tutorial for each of these steps that I've described. You can also download the results folder right here with all of the results that TIMEOR has generated and more. Thank you for your time. Enjoy.